Hi everyone, welcome to the Annals of Botany lecture at Botany 2020. My name is Jeff Karen. I'm the North American editor of the Annals of Botany. We've been proud to sponsor this lecture series for the past 18 years. Founded in 1887, the Annals of Botany is the world's oldest plant science journal. We publish novel and substantial research in all areas of plant science. And we're especially well known for papers in the field of plant reproductive biology. It's truly an honor to introduce today's speaker. Professor Rod Peakall is internationally known for his innovative research on sexually deceptive orchids. His elegant experiments have demonstrated that floral volatile compounds can control pollinator specificity, pollinator switching, and speciation. Rod's research team has found strong evidence that pollinator-mediated isolation can be a primary reproductive barrier in sexually deceptive orchids. Not only is Rod a keen observer of nature, he is also a gifted nature photographer and videographer. His talk today will showcase some of his stunning videography. Rod received his PhD from the University of Western Australia and then completed postdoctoral research at Macquarie University and Rutgers University. Since 1993, Rod has been a professor at the Australian National University. He has trained more than 25 PhD students, as well as hosted about 15 postdocs. Rod has published more than 170 papers, including many recent papers in the Annals of Botany. A few years ago, he was a co-organizer of a special issue of Annals of Botany on pollinator-driven speciation. Rod is also well known as the first author of the widely used population genetics software package, Genelex. His talk today is entitled Sexual Deception in Australian Orchids, Insights on the Evolution of Extreme Pollinator Specialization and its Reversibility. Please welcome Rod Peakall.
Hello everyone, thank you for joining me. It's a very great honour and privilege to be invited to give the Annals of Botany special lecture for Botany 2020. A very special thanks must go to Jeff Karen, both for the invitation and for his generous introduction to this talk. Thank you, Jeff. Thanks also to the Annals of Botany. It's been my pleasure to publish a number of papers in this journal on a range of topics over the last few years, and I will of course briefly highlight some of these. It's also very important for me to acknowledge my ongoing collaborators who are based at 3 Australian universities and the University of Michigan. Without their vital contribution, there would be no story to tell. Finally, last but by no means least, I acknowledge the Australian Research Council and thank them for their generous funding over so many years. To set the context, it may be helpful to remind you that more than 85% of the flowering plants depend on animal pollinators, with these plant-pollinate interactions varying along a continuum from generalised to highly specialised. In generalised cases, such as illustrated here by Asclepius, multiple different types of flower visitors, including bees, wasps and butterflies, are legitimate pollinators. In the mid-range of the continuum, the plant is specialised on a single functional pollinator group, such as in this case of Mimulus ringens, which is pollinated by several different species of bumblebee. Of course, one group of plants where moderate to extreme specialisation seems to be the norm is the Orchidaceae. Coincidentally, in a review just about to come out in the Annals of Botany on orchid conservation, we've compiled a summary of the estimates of the frequency of specialized pollination across various orchid floras, revealing a modal value of 1 is common. That is, having a single species of pollinator for any given orchid species is the most common finding at both regional and global scales. Thus, while orchids exhibit the same pollination continuum, they strongly tend to occupy the far right. Perhaps a pollination strategy with the highest incidence of extreme specialization, indeed where a one-to-one relationship between orchid and pollinator species is the norm, at least at the site level, is pollination by sexual deception. A simple definition for sexual deception is pollination by sexual mimicry. In the orchids at least, the mimicry includes both the chemical mimicry of the sex pheromone of the female of the pollinators, as well as physical mimicry. As I've just noted, extreme pollinator specialization is a norm. However, it is worth noting that this extreme specialization is perhaps an inevitable consequence of evolving sex pheromone mimicry for pollinator attraction, rather than a consequence of being strong selection for extreme specialization per se. It's evident from this map that sexual deception is a worldwide phenomenon, with independent evolution on 4 continents. It's also worth noting that while orchids dominate the known examples, single cases each are known in a daisy and an iris. The strategy is perhaps notably absent from North America and much of Asia. While I expect the absence in North America is real, for Asia it's most likely merely overlooked. The panel on the right illustrates some multiple orchid genera employing this pollination strategy among the Australian orchids. In Australia, sexual deception has multiple origins across the terrestrial orchids, and it involves the sexual exploitation of males from 5 families of Hymenoptera and 2 families of Diptera. To date, most of our research is focused on 2 independent radiations containing sexually deceptive orchids that exploit male thin-eyed wasps as their pollinators. Some 70 species are represented in the clade containing Chylogotis and Drachea, shown on the left. At least 150 Calodenias out of some 250 species are also sexually deceptive in the clade, shown on the right. So how does this pollination strategy of sexual deception actually work? Or at least how does it work in Australian thin-eyed wasp-pollinated orchids? The thin-eyes are a diverse southern hemisphere group of parasitic wasps in their own subfamily that reaches its highest species diversity in Australia. All species exhibit moderate to extreme sexual dimorphism, with the females wingless, the males winged. Here you can see a close-up of the female in the top left and pairs in copula to the right and below. Now let's take a look at a day in the life of a female thin-eyed wasp. Here's that flightless female. She's just emerged from underground. And she's now in what we call a calling position, releasing sex pheromones to attract a mate. It doesn't take long for the male to find a female and to pick her up. We'll watch that again in replay. The pair in copula then proceed to a food source, usually a nectar source, and if you watch closely you can see the male here is feeding the female. The male subsequently drops the female back home where she burrows in search of larvae on which she will lay her eggs. Perhaps the most compelling example of the extraordinary exploitation of the thin-eyed wasp mating system is illustrated by Drachea orchids shown here in this video. Now that we better understand the mating system of the pollinator, it's time to ask, how is this interaction achieved? In plain English, we can of course ask the simple question, is it good looks, good chemistry, or both? Now when you compare the visual similarity between this orchid and the female of its pollinator, it's certainly tempting to speculate that it's all about good looks. However, let me convince you otherwise. In this experiment, I'm dispensing synthetic copies of the sex pheromone of the wasp onto a little black bead, at best a very rudimentary mimic of the female, and as we can see, the bead with chemicals elicits strong sexual behaviour, confirming the importance of long-distance chemical attraction. I'll come back to the actual chemistry in this particular case later. Clearly, the chemistry of the sex pheromones holds the key to how pollination by sexual deception works. Therefore, commencing in 2001 with an international research team, we set out to unravel the chemistry of these interactions. The journey has revealed many exciting and unexpected discoveries, and there still remains much to learn, even after almost 2 decades of research. The most I can do in this short time is to give you a snapshot of some of the highlights. To get to the chemistry of these interactions, we employed well-established procedures. I'll omit the details here, other than to note 3 things. First, one important step is to ask the pollinator what it can smell. That is done via the process called GCEAD, short for gas chromatography with electroantenography detection. Second, productive partnerships between chemists and biologists are absolutely critical. Third, there is lots of work for biologists to do nonetheless, not least in performing the final field confirmation step. In this video clip, we can see the bioassay convincingly demonstrates that a single synthetic compound elicits very convincing sexual attraction of an orchid pollinator that we can see here carrying orchid pollinia. Now, what were our expectations when it began? First, as was already known in moths and bees at the time, we predicted that the wasp sex pheromones would involve blends of compounds from a common chemical system. For example, across the hundreds of moth pheromones known, 75% of all cases involve simple, biosynthetically related, so-called type 1 compounds. The majority of the remaining cases are drawn from a pool of biosynthetically related type 2 compounds. We also predicted that the orchid semiochemicals used for pollinator attraction would chemically match the sex pheromone of the wasps. Finally, we anticipated that the specific attraction would be achieved via the use of unusual blends of otherwise common compounds. Furthermore, with more than 1,700 floral scent compounds already known, we fully expected that the compounds involved would be drawn from this large set. Now, our exploration of the chemistry of sexual deception began with this diminutive little red and green orchid, Chylogotis trapeziformis. We focused on this species in part because both the flowers and the pollinator were accessible literally across the road from the Australian National University in the very centre of Canberra. Now, pollinator behaviour at the flower is more or less typical for that of many sexually deceptive orchids. Here we can see the male attempting copulation, probing with the genitalia exposed on the very tip of the labellum here. Ultimately, during this attempted copulation, the wasp's thorax contacts the stigma, removing stigmatic secretions, which then act as a glue to securely remove the pollinium. Subsequently, a visit to another flower results in pollen transfer, and the process of pollination is complete. Here we can see that pollen left behind very clearly on the stigma. To cut a long story short, and due to some brilliant chemistry by Whitko Frankie's group in Germany, we identified the first thinine wasp sex pheromone in 2003. Quite unexpectedly, this single component sex pheromone represented an entirely new class of natural products. Furthermore, The same identical compound was the semiochemical used by the orchid to secure the male wasp attraction. Now, throughout this talk, I will be using stick drawings like that shown on the left. For those of you who may not think too much about chemical structures, the lines in the stick drawings represent carbons, with other compounds except hydrogen clearly marked. By the way, we called this first compound Chyloglotin 1 after the orchid genus Chyloglotis in which it was first found. In rather quick succession, we were able to discover a family of 6 different Chyloglotones which were involved in orchid pollinator attraction. We were also able to experimentally confirm the chemical basis of pollinator specificity. For example, in this series of choice assays, we presented 3 different pollinators of 3 different orchids with a choice of either Chyloglotones 1 or 3, or a 1-to-1 blend of the 2 compounds. The pollinator of Chyloglotis trapeziformis at the top, whose sex pheromone we know is Chylo 1, barely responded to the 1-to-1 blend and never to Chylo 3. Conversely, the pollinator of Chyloglotis affinity Jeansii, down the bottom, only responded to Chyloglotin 3. The orchid pollinator pair in the middle is of interest since some responses of the pollinator were observed to the blend, but not to the alternative, suggesting that cases like this, where the pollinator can respond to a blend, might provide the bridge for Chyloglotin transitions between species. In other cases, we've indeed shown that the specific blend of 2 Chyloglotones is the pollinator attractant and the basis for pollinator specificity. These and many other similar experiments convincingly confirm that chemistry controls pollinator specificity in these cases of sexual deception. Now, additional bioassays like those illustrated in the last slide, involving a range of orchids, different pollinators and different synthetic Chyloglotones, have not only revealed the chemical basis of pollinator specificity, but they've also revealed compelling evidence that these chemical traits represent axes of variation under strong pollinator-mediated selection. For example, we've seen evidence for strong directional selection for a single compound, as shown in the previous slide. Conversely, we've also uncovered evidence for strong stabilizing selection for a specific blend, in one case for a blend in the range 1 to 10 to 1 to 50, involving Chyloglotones 1 and 2. We've also obtained evidence for selection for more compound emission. In all these cases, the response measured, which indicates the direction of selection, has been increased attraction, increased lands on the flowers, increased attempted copulation, and often the increased duration of attempted copulation. We've further demonstrated that the increase in attempted copulation and increase in the duration of attempted copulation serve as a very good proxy for fitness. Now, in several studies, we've also demonstrated that variation in Chyloglotin chemistry defines species boundaries and underpins very strong pollinator-mediated reproductive isolation. Time doesn't permit me to walk through the details, other than to note that a corollary of chemistry defining species boundaries is that orchid species need not be morphologically distinguishable. Indeed, we've uncovered several cases involving cryptic sympatric taxa that cannot be reliably distinguished by morphometric analysis, yet are categorically identifiable by their floral odor chemistry, and this has been confirmed by genetic analysis. In a further interesting and utterly unexpected twist that we published in several papers in the Annals of Botany, we discovered that the production of Chyloglotones is dependent on UV light, although the precise mechanism remains elusive. We further predict that this entirely new and unexpected biochemical reaction might also occur in taxa other than these orchids. In further and ongoing studies to elucidate the biosynthesis of the Kylogodones, we've been using the UV dependence as a tool. We've also very recently demonstrated in another Annals of Botany paper that gene duplication and selection may hold the key to the evolution of Kylogodones. Now let's move quickly beyond Kylogodus and Kylogodones, because they are by no means the end of the chemistry story. Commencing in 2009 with new chemists as collaborators, including a crucial postdoc in my lab, Bjorn Bowman, and with Gavin Flamati, based at the University of Western Australia, we began to uncover more extraordinary chemical diversity. Here are some of the target species we've investigated. Let's first take a quick look at the case of Chelidenia crebra. In this orchid species, we discovered that a set of 4 related sulfurous thiophenol compounds were the semiochemicals involved in pollinator attraction. 3 of these compounds were previously unknown in nature. A combination of additive and detractive bioassays further revealed that a 10-to-1 blend of 2 compounds was absolutely essential. The same compounds were confirmed as constituents of the sex pheromone of the female of the pollinator. For the next case of Chelidenia plicata, I would like to share with you the video abstract accompanying our current biology paper, since this is perhaps the best way to compress the outcomes of one of our most exciting studies into a 1.5-minute segment.
Sexual deception is an intriguing interaction in which the plant secures pollination by sex pheromone mimicry. The chemicals known to be involved consist of related compounds. Here we can see a male wasp attempting to mate with the orchid. This Zelloboreus species is the only pollinator. Unexpectedly, 2 chemically distinct compounds hold the key to this interaction. Field experiments confirm the biological activity of these 2 compounds. The methylacetophenone has never been found in flowers before, while the monoterpene, citronellol, is well known as a floral volatile. Given that geraniol and citronellol differ only by a double bond, a one-step synthesis has been predicted. However, we show that citronellol biosynthesis is a multi-step process. This study has uncovered complexity at the chemical and biochemical level, revealing that in this case, sexual deception is achieved by co-opting 2 distinct biosynthetic pathways.
Now, in subsequent work, we further demonstrated just how finely tuned the chemistry of this orchid-pollinator interaction involving Chelidenia plicata and its Zelloborea pollinator. You may have already noted from the video clip that the optimal ratio of the 2 active compounds was 1:4. What we didn't tell you is that there are 2 enantiomers of the citronellol and 10 structural isomers of the acetophenone, all being possible alternatives to the active compounds. In fact, the chemist had to make the full set of 10 isomers to ensure we'd identified the correct natural product. In the field, I conducted bioassays to test the activity of alternative enantiomers, S&R, and all 10 isomers and 1 additional analogue. Remarkably and unexpectedly, given the very subtle chemical differences between some of the acetophenone isomers, only the 2 natural products worked. Thus, in this case, the complexity of sexual deception requires not only an optimal blend of 2 compounds from biosynthetically different pathways, but precisely these 2 very specific compounds. Of course, it remains a mystery as to how, in evolutionary terms, orchids can intercept and co-opt the private communication signals of their wasp pollinators in such a precise way. Clearly, Chelidenia plicata demonstrates another axis of chemical trait variation under strong pollinator-mediated selection. In this case, we've accumulated very good evidence for strong stabilizing selection for an optimal blend involving two unrelated compounds. Drachia species represent another interesting case study in that the genus is very closely related to Chylogotis. Yet, at least in Drachia, rather than using chylogodones, specific blends of nitrogenous pyrazines are involved, including new to science hydroxymethylpyrazines. The case gets even more interesting. In fact, Drachia micrantha represents the second case of complex sexual deception involving two unrelated types of compounds. Here, pollinator attraction requires a specific blend of two pyrazines and a novel type of compound we've called Dracholide, after the genus Drachia, in which it was found. Incidentally, the Dracholide shown here will become Dracholide 1 because we already know that related Dracolides are involved in pollination in other Drachia species. It will now be evident that contrary to our original chemical prediction of a common chemical system, we could not have been more wrong. Instead, so far, we've uncovered 5 very different chemical systems that are involved in sexual deception, and we know there are even more. And these are just the cases involving thinine wasps. This chemical diversity is truly extraordinary. Furthermore, the emerging discoveries of mixed chemical systems has been even more surprising, since sex pheromone systems involving combinations of biosynthetically unrelated chemicals appear to be rather rare in nature. A second chemical prediction that the compounds involved would come from the set of 1700 plus known floral volatiles is also not supported, barring the exception of the monoterpene in Caledonia plicata. Instead, many new to science and even entirely new classes of natural products are involved. Perhaps the only chemical prediction that is fully supported so far is that of precise chemical match between the orchid semiochemicals and the sex pheromone constituents of the pollinator, although we've accumulated evidence that analogues are also possible, and we fully expect that this hypothesis will not hold in all cases. So I hope I've been able to convince you that one thing is clear, and that is that pollination by sexual deception takes chemistry to work. At least that is true in the Australian sexually deceptive orchids we've investigated so far. Now that we've established beyond doubt this critical role of chemistry, let's turn to a new question. Does morphology matter? Certainly when looking over this collage of some of the sexually deceptive orchids of Australia, one cannot help but notice that these look more like insects than flowers. With Marina Stiega, who joined my lab on a brief but very productive postdoc, we set out to explicitly address this question, does morphology matter? To his credit, the first of his 2 papers was featured in Nature's Research Highlights of the Week in September 2015. Now, typically orchids with different morphologies will have different chemistry. This poses a big challenge when investigating the role of morphology because any pollinator behavioural differences among species could be entirely due to the different chemistry. Therefore, to avoid this potential confounding effect, we identified an ideal pair of study species. Chylogotis trapeziformis, the species in which we first cracked the chemistry, and the allopatric Chylogotis valida, which attracts its own pollinator but via the same chylogoine 1. Thus, shared chylogodone chemistry underpins the chemical mimicry in both these cases. Conveniently, these 2 species also have rather different floral morphology, with pollination occurring in contrasting orientations. In the first case, the pollinator orientation is forward, while in the second, the pollinator orientation is reverse. This allowed us to predict a priori and to subsequently test the different expectations for each orchid species. For example, due to these different orientations during pollination, the mechanical fit predictions differed between the 2 species. In the first species, we predicted labellum length would correlate with the male pollinator length, whereas for the second species, the distance we called the calli length should be correlated with the pollinator abdomen length. These and other related predictions were indeed supported. In a further extension on the mechanical fit hypothesis, published in the Annals of Botany, Marinus experimentally manipulated the labellum length, either shortening or elongating relative to the average size. In baiting experiments with these modified flowers, the duration of attempted copulation, a confirmed proxy for fitness, was significantly shorter for both treatments. Additional evidence for pollinator-mediated selection was provided by phenotype selection analysis. Collectively, the results provide strong evidence for pollinator-mediated stabilizing selection for floral size in Chylogotis. Returning to the evidence for morphological mimicry, the mimicry predictions also differed between the two orchid species, with the evidence providing support for both matches in the essentially achromatic colour of the female and the orchid, and in the relevant size matches. In summary, these experiments have uncovered additional axes of floral trait variation under pollinator-mediated selection in sexually deceptive orchids. These include both morphological traits associated with mechanical fit to the male pollinator and traits associated with female mimicry. With a former postdoc and ongoing collaborator, Ryan Phillips, another strategic target species for investigating the role of morphology has been this beautiful orchid, Calodenia pectinata. In yet another entirely unexpected discovery, Calodenia pectinata sexually attracts the same pollinator as Drachea livida. The pollinator, Zasperlothinus nigripes, with male and female in copula, is shown in the middle of this figure. At face value, this finding would seem to argue strongly against the importance of any morphology, but things are a bit more complicated. One very important difference between these two orchid species is that chemical and physical mimicry is tightly coupled in Drachea livida. Here the insectivorm labellum is both the source of the chemical attractant and the physical mimic of the female. By contrast, in Calodenia pectinata, the sepal tips are the sole source of the chemical attractant, with the maroon labellum at best a rudimentary colour mimic of the female wasp. The consequences of this uncoupled physical and chemical mimicry in Calodenia pectinata is clear in the video that follows. Now take a close look. What are we seeing happening here is attempted copulation with the sepal tips, the source of attractant, not with the labellum. Of course, contact with the labellum is essential for pollen removal and indeed for pollination, but it appears that attempted copulation is not required. Rather, we now believe that on the labellum there's an instantaneous switch to food-seeking behaviour, perhaps triggered by floral scent compounds. Now in Calodenia pectinata, our experiments confirm that if we move dissected sepal tips, which are the source of attractant, under the labellum, then attempted copulation shifts to the labellum. What is more, copulation rates and duration go up dramatically, confirming that sexual behaviour is indeed enhanced when chemical and physical mimicry is coincident. The results from additional experiments hint that red-coloured labella are favoured over white-coloured ones, although more work is needed to fully tease out the role of labellum colour in sexual attraction. So what about the evolution of sexual deception? As an extremely specialised pollination strategy, one of the big evolutionary questions we've begun to consider concerns the reversibility of sexual deception. As you may well know, extreme specialisation has long been predicted to be an evolutionary dead end. Furthermore, a molecular basis for the irreversibility of some pollinator transitions is well established. For example, during transitions to hummingbird pollination in North American cases, the flower colour shifts from blue to red are repeatedly due to loss of function mutations, which have then been followed by gene degeneration, likely rendering the trait irreversible. However, a number of studies, some shown here, indicate that reversals from extreme specialisation is possible in some cases. Now, the genus Calodenia, with more than 300 species, is a prime candidate for exploring this and other questions on the evolution of sexual deception. In fact, it's currently the only known sexually deceptive genus in the world to also contain species using other pollination strategies. Now, while cases of selfing and food reward by nectar are very rare, food deception is a common strategy, and it's long been considered that sexual deception arose from food deception in this genus. Indeed, to this date, all publications, including our own, have at least implicitly assumed that sexual deception is irreversible. Now, one of the key impediments to testing this hypothesis of reversibility of sexual deception has been the inability to build a well-resolved phylogeny for Caledonia and its outgroups. In an attempt to overcome this issue, we've taken advantage of next-generation sequencing tools and built our own custom sequence capture design. Consequently, we now have some 1.5 million base pairs of sequence spanning 1,500 loci for more than 300 samples. Now, given a phylogeny with 300 samples is far too complex to show here, I've taken the liberty of showing you an illustrative phylogeny. Let me walk you through some of the key findings from our ancestral trait reconstruction. First, as shown here, the basal taxa are food-deceptive taxa. The first cases of sexual deception occur on this node and on the parallel node here. Now, it's worth noting here that our ability to resolve these nodes is only now possible for the first time. Furthermore, as we step through the phylogeny, node after node contains sexually deceptive species. The oldest example of sexual deception appears to be represented by this taxon, followed by this one. It's thus evident that sexual deception underpins the two main radiations of Caledonia. Now, that's not the end of the story, because as we drilled into the phylogeny in a little more detail, we discover that in clades C4, representing approximately 70 species, and clades C5 to C9, representing more than 200 species, independently arising out of these two radiations is a repeated convergent evolution of white and other colourful food-deceptive spider orchids. This new phylogenetic evidence confirms that extreme specialisation is indeed reversible in some, but not all clades. Not surprisingly, this entirely unexpected outcome has literally forced us to rethink the current published hypotheses on the evolution of sexual deception, and to seriously think about possible mechanisms for the reversals that we're seeing. Now, it's worth zooming in a little closer and reminding you that not all of the sexually deceptive clades show evidence of reversibility. Interestingly, the irreversible clades C5 to C7 contain species with highly reduced flowers and strongly insectivorm structures on their labella. Many of these really do look more like insects than flowers. Some even show remarkable convergence to the flowers of Chylogotis and Drachea, which, as you recall, belong to a separate independent clade of sexually deceptive thin-eyed wasp-pollinated orchids. Incidentally, it appears that sexual deception is irreversible in that clade. Now, given the very compelling evidence I presented earlier for strong pollinator-driven selection for chemical and female mimicry, how is escape from such strong selection even possible? The short answer may come from clues first indicated to us in the case of Caledonia pectinata. Recall that in this case only the sepals are the source of the sexual attractant. Now, intriguingly, we've since discovered that uncoupled mimicry is not uncommon. In fact, we have experimentally confirmed that one or more species in the reversible clades exhibit uncoupled chemical and physical sexual mimicry. Therefore, we predict, although have yet to confirm, that uncoupled mimicry is the first step, or the pre-adaptation, as it were, for subsequent reversal. In brief, our working hypothesis is that with coupled mimicry and its associated pollen removal during sexual behaviour, strong directional or stabilising selection for sexual mimicry is unavoidable. This likely means reversals out of sexual deception are also impossible. On the other hand, uncoupled mimicry, potentially allows a switch in pollinator behaviour, with pollen removal now occurring during food seeking rather than sexual behaviour. Such cases might be better described as pollination by sexual attraction. We are now beginning to suspect this is a widespread and stable pollination strategy across the larger and more colourful Caledonia spider orchids. In those cases where pollen removal is no longer dependent on sexual behaviour, we predict that selection away from mimicry is a real possibility, potentially enabling the evolution of food-deceptive and even food-rewarding pollination strategies from sexually deceptive ancestors. In the context of these hypotheses, we've now identified a set of strategic study species to explore the ecological, chemical, and molecular basis of pollinator shifts and pollinator transitions. Funding permitting, we hope to begin work on these topics very soon. Let's turn now to the conclusions. I hope that it will now be self-evident that the fascinating, perhaps even bizarre pollination strategy of sexual deception is characterized by unexpected chemical and morphological complexity. The plant species involved are subject to strong selection across multiple axes of floral trait variation. In fact, given the extreme pollinator specialization, sexually deceptive plants perhaps are among the most compelling examples of pollinator-mediated selection. Furthermore, at least among Australian orchids, sexual deception underpins multiple radiations. It is also evident that for some clades at least, sexual deception is not an evolutionary dead end, with reversals possible. Certainly, investigating the interplay between plants and pollinators has revealed extraordinary and perhaps somewhat overlooked potential for evolutionary capacity and flexibility. I dare to predict that escape from extreme specialization may be far more common in nature than we currently think. Furthermore, I suspect that this untapped evolutionary flexibility may hold the key to the future survival of many species facing pollinator declines and environmental change. In this sense, I hold some optimism for the resilience of nature, despite the odds and the untold scale of human-induced change. I would also like to stress this one last point, that is, that exciting discoveries and novel insights come from multidisciplinary research. Yet, all too often individuals, organisations and funding agencies overlook the extraordinary potential value of multidisciplinary collaborations. In the final few moments, may I put on my mentor's cap for a moment and reflect on some lessons I've learned as one of the older guys. In particular, I think these lessons will be relevant for younger scientists, be they graduate students, postgraduates, or other early career researchers. First, never say never. In the story of sexual deception I've just told you, and indeed in other different studies across my career, we've made scientific discoveries that we never expected. These discoveries often revealed far more complexity than our initial hypotheses, sometimes even reversing our scientific understanding. It follows to expect the unexpected. I also urge you to take advantage of lucky breaks to exploit serendipity. This might be as trivial as taking advantage of a rare combination of seasonal conditions to study a new plant-pollinator interaction, or it might be forging new collaborative partnerships that happen when you accidentally cross paths. I would also stress the importance of being patient. Scientific breakthroughs often take time and patience. In particular, working with plants and pollinators in the wild can be really challenging. Such studies certainly demand patience over multiple years of fieldwork. Finally, above all, have fun. With those conclusions, I close with this more detailed acknowledgement slide. I would also like to thank Jeff Curran again for his invitation to present this special lecture. I'd like to thank the Annals of Botany for their sponsorship, and to thank the organisers of Botany 2020. I look forward to taking your questions.